Hi, everyone. Welcome to this month's RNA Collaborative Seminar. I'm Maddie Sherlock. I'm an assistant professor in the Molecular Cell and Developmental Biology Department here at the University of California, Santa Cruz. And it's my pleasure to introduce our first speaker. Todd Lowe is a professor in the Biomolecular Engineering Department here at UC Santa Cruz. He's also affiliated with the Genomics Institute and the RNA Center. He received his Ph.D. from Washington University in St. Louis, where he worked with Sean Eddy, and then completed his postdoctoral training with David Botstein at Stanford before joining the UCSC faculty in 2001. And since then, his work has integrated computational and molecular biology approaches to characterize known non-coding RNA genes in a wide range of organisms from humans to extremophilic archaea. His lab has developed computational pipelines and methodology, including high-throughput sequencing techniques to study non-coding RNA structure, function, regulation, and modifications, especially in tRNAs and tRNA fragments. Today, he'll be telling us about some of his recent work on human tRNAs. And before I hand it over, I wanted to let you all know that we will be hosting a conference on our campus this August. Registration is now open for the UC Santa Cruz RNA Futures meeting, which will have some fantastic keynote speakers and also includes an optional pre-meeting workshop, which will be led by Todd as well as Angela Brooks on both long and short read RNA-seq. Todd will have the meeting flyer at the end of his talk. We'd love to see you here. Um, Finally, please remember to put your questions in the Q&A, not the chat, for me to read after Todd's talk. And with that, I will turn it over to Todd.
Thank you, Madeline. Start my slideshow. Okay, I assume you can see my screen. Yeah, okay. All right, thank you everyone for joining us. I have been working for quite some time with tRNAs. I started as a graduate student. I helped write a program that was used to identify the tRNAs in the human genome as well as most other genomes. And these days, though, it's getting very exciting because there's a ton of functional genomic data that's coming together with human variant data and lots of different stories that are starting to come to light. So hopefully I can share some of these with you. Okay, so this is the standard stock slide. tRNAs are ancient. They're very old. They've been around. People have felt like they understand what tRNAs have been doing for many years. Their central role in translation. However, there are a lot of new roles that have come to light in terms of fragments of tRNAs that become regulatory molecules, as well as other features of tRNA genes themselves where they can become chromatin insulators, actually. So they actually have barriers to activation or repression, and they can separate out different regions of the genome from a regulatory perspective. So tRNAs continue to give us new surprises. This is just a quick overview of the loci where the tRNA genes are in the human genome. There are about 619 total, but there are only 429 that we call the high confidence genes that have features that are strong, we believe are necessary for being functional. And there's some molecular data to support those classifications. So chromosome 1, for example, has a lot of different tRNAs. One notable chromosome X, only has 3 or 4 tRNAs, only one that we think is functional. Y doesn't have any. And so there are a lot of different neighborhoods that we believe are involved in different functions for these tRNAs. And now from a transcript perspective, there are sort of 3 different subclassifications of tRNAs that it's important to understand. Isotypes, of course, these are the tRNAs and the amino acids that they're charged with. Isoacceptors, these are all charged, for example, with alanine, but they have different anticodons. And so functionally, these are all very distinct and important. And then an extra layer that I'm going to be focusing on a lot for this talk is sort of unique transcripts or isodecoders where they have the same anticodon, but the sequences of their tRNAs are actually different. And it's still somewhat a mystery of why we have multiple different versions of these. And our current models, or the models have been used traditionally, have not really spoken to these roles. This is just an example. It's a screenshot from the genomic tRNA database where we house alignments and all the information about the tRNAs. Here are two different asparagine tRNAs where there's just one change between these two. These two different forms, ASN GTT1 versus 2. This first letter is sort of the transcript or isodecoder number, and the second number here is the copy number of that in the genome. So if it's -1, it's the first copy, and if this number is higher, those are other copies in the genome. But I'll talk more about that later. So there are some cases where there are many differences between these different isodecoders. Serine CGA, both of these have CGA anticodons. However, they have very different sequences in their acceptor stem and at multiple other places. That in some cases, some of these might be sites where there's a modification that's on one and not the other. So these can be sources for variation that may have very important functional distinctions. So something else I like to go over is the total gene count of tRNAs does not equal their complexity or their functional complexity. An example of this is where we have budding yeast. There are 275 total genes. In human, we have about 429 high confidence genes. However, if you look at the unique transcripts that those genes produce, there are only 55 in yeast, basically just one version of each isodecoder. So there's just one with each of the different anticodons, but many copies of those. So there's a lot of redundancy, but not a lot of sort of diversification. In humans, about half of the tRNAs are unique. And so this is, again, very interesting and something that we want to understand better. This is a display to sort of overwhelm you. These are all of the human arginine tRNA genes. And you can sort of see just, in glancing, there are lots of little differences between these. But this is sort of the extent of our functional understanding of the differences between them. The sequence differences. There are not a lot of functional differences ascribed to these until fairly recently. And so I'm going to be talking to you about how we've started to tease apart function for how each one of these actually look like they may have different roles and different importance to translation and other roles. So how do we study hundreds and hundreds of genes? Well, tRNA sequencing. There are new technologies for sequencing tRNAs where the tRNAs are, because they're very highly modified, previous RNA-seq methods did not work very well. So only in the past 10 years do we really have good transcription data for tRNA sequences. There's also a lot of epigenomic data out there for chromatin state and Pol3 ChIP-seq, which tells us the activity levels of individual loci. And then we do a lot of evolutionary analysis between different species to show what things are changing and what things are staying the same. So here's sort of the superstar in terms of human tRNAs. This was actually identified and studied in a pretty amazing paper way back in 2014. And it showed that there's one particular arginine UCU tRNA where when there's a mutation in this that's combined with a mutation in the ribosome release factor, it causes severe neural degeneration. And so Sue Ackerman has studied this for many years. And what's surprising is that this particular one tRNA is very abundant in the brain and not abundant in any other tissues. And yet we have 4 other copies of this ARG-UCU that do not cover or are not redundant with this one. So this one has unique features that it's still not completely clear is really important for neural and brain function. So this is the first clue that the individual tRNAs, actually, if you have disruption of their function, the other tRNAs in the isodecoder group or isoreceptor group are not necessarily going to be able to cover for them. Here's a profile of that ARG-TCT4. It is the previous slide was talking about mouse. This is expression data, tRNA expression data from GTEx samples, which are human normal samples. And you can see that the ARG-TCT4 is high and as expression in brain cortex, but it's virtually absent in testes, liver, whole blood, and this is actual data that we've collected ourselves. Also, from a sort of an oddly conserved genomic perspective, these are all different species across vertebrates. And this gene, if you look at, is nearly invariant, which is really, really unusual. So there's something very special about this one, and it is definitely at the very top of the list in terms of hyper-conserved tRNAs, for which we do not fully understand why it is so highly conserved and how it functions differently in neurons. But with that one, you can then start to say, well, are there other tRNAs that are tissue biased or tissue specific? And this is data that's from a paper that we have submitted to bioRxiv and that is in the process of being submitted to a journal. It is a tissue atlas of across 19 different tissues for mouse. And so these are different tissues across the bottom, and these are different tRNAs across the right here. And I don't have time to go into sort of all the little minutia of this, but you can see here that for brain, there are quite a few tRNAs that the darker the color here, the more tissue bias that are very biased for brain tissue. But there are also other tRNAs that are biased for other tissues. And right in here, I have the arrow pointing at this ARG-TCT4, which is highly brain biased, and it's hardly expressed in any of these other tissues in mouse. But there are other tRNAs that are highly expressed and fairly uniquely expressed. But a lot of them are expressed in other tissues, but just not nearly as highly. So this is good evidence that there is specialization of tRNAs. Okay, so about half of the mammalian tRNAs are multi-copy. So if we want to try to understand the basis for the regulation of this tissue specificity, it's very difficult because multiple loci in the genome are producing the same transcript. So to the rescue, we can go to the chromatin level. So way back in 2015, there was this wonderful study where they looked at 111 different cell types, both mature tissue types, fetal, and cell lines. And across all those, they basically profiled ChIP-seq data, which gives information when combined together for the activity level of any particular gene in that position. And so the amazing thing about this study is they completely ignored non-coding RNAs and especially tRNAs. And so we went into this data and then mined it to look to see which tRNAs are active at the gene level, not necessarily just at the transcript level. And this is what we found. We were pretty astonished to find that, and I'll first explain this to you. So these are the different sample types across the top. These are 120 different sample types. And along this axis, each one of these rows here is a different tRNA. And so if every single tRNA was active, this whole thing would be red. This red state is basically the red and orange are transcriptionally active states for that part, the chromatin. And so this is a stand-in for the fact that the chromatin is open and it's open for business. And so it's pretty surprising that only about 19% of all of our tRNA genes are constitutive. The rest are either on in some tissues and not others. Some are heavily polycomb repressed and are really only on in development and under special growth conditions. There's another class that are overlapping Pol2 genes, mainly in their introns. And then there's a whole very large class of silent genes down here, where even across all these different tissue types, there was no evidence or very, very little evidence that these are actively transcribed. And so these we've been calling silent. So these are these epi groups that I will start to refer to regularly, from highly active to partially to only on under special growth conditions, and then mostly silent. And so this already tells you that if there are 5 copies of a gene, they may not all be active. They may be in different states, and so we don't have as high a redundancy in our gene set that we believe. So here, zooming in on one particular set of tRNAs, the leucine TAA or UAA tRNAs. There are four tRNAs, oh, sorry, five of these, and they all have pretty different programs. So one locus, this Lu TAA11, this one is highly transcribed. It's read through all of these different cell types and tissue types. This Lu TAA31 is on in most of them, but not all. And then we have some that are two that are only on in stem cells and rapidly growing cells. And then one of them is only on or doesn't seem to be on at all. So these are really diverse programs that most people are not aware of. And this is all from the chromatin IP data. Now, what does this mean? Well, we can start to look at the sequence features that are different between these, and these are all different. They all have different numbers here, so they are all different versions of the transcripts. So then if you look more closely, one of them, the transcriptionally constitutive one, or one of them, is this Lu TAA. What I have here in green are basically the determinants that allow for the identity elements that allow for charging, very specific charging that all tRNAs have, that the tRNA synthetases need in order to accurately charge the tRNAs. And what's fascinating is that this other tRNA that we know is expressed, but it's only expressed under very specialized conditions. Several of the positions that normally would be AU or GC over here are different. So this implies or suggests that these are probably charged at a different rate or at a different efficiency. And so already you can infer that there's actual real functional difference between these. Again, the one that's sort of a housekeeping version and one that's only turned on under specialized conditions. Here's another example, and I will go through as as many as I can, maybe two or three more. But this is cysteine, where in this case we have actually four different copies of the same gene. So 2.1, 2.2, 2.3, 2.4. You can see these are the different genomic locations. Three of them are on chromosome 17, and one is on chromosome 4. They have four different programs, but we have three different types here. We have one that's constitutive, two that are variable, one that's much more variable than the other, and then one that's mostly silent, but you can see is overlapping actually some kind of Pol2 transcript, either a link RNA or a protein coding gene. So together, because these all produce the same mature transcript, this is the result. And so you can see that this can be very difficult to figure out, okay, why do we have this particular expression pattern when you have contributions of four, probably more likely three different loci? This is very common. And so this is what makes it difficult. This is why we don't know already sort of the programs for these tRNAs, but they are unique and they are, I think, important to disentangle. So the thing about these is they all have identical A and B boxes. tRNAs have internal promoters, the A and B boxes. So these guys all have the same promoters. The fact is they're different places in the chromatin. And so prior paper from Genome Research we published in 2020, we looked at the context of the genes. So we found that CPG islands nearby, being near other sets of genes, were indicative of active tRNAs, and ones that did not have these poly-T terminators at a sort of normal distance, or that had suppressed CPG and other features, are signs of inactive tRNAs. And so genome context matters a lot. Here's a couple examples of Lu CAG that's really fascinating. So here's the tRNA here, the clover leaf, and I'm talking about this one particular 667 base pair right here. And so we have Lu CAG1 and 7 copies of this, identical copies, and then 2 identical copies of Lu CAG2, 2.1 and 2.2. And the only difference between these is that this 667 is a GC in the Lu CAG1 copies, and it is a GU in the others. And Um, you might think, oh, these are just mutations of some kind. But then if you look across evolution, which is another way that we look at these, this is actually a profile from our tool tRNA-Viz. Anyone can use this. You can put in the particular position in the tRNA and then the particular clades and subset of species you're interested in. And you can see here that insects and nematodes, they have GU position, GUs or UGs for the most part at this particular 667. But primates and mammals, I don't have space to show mammals, have both versions. They all have both versions. So they picked up sort of this some kind of diversification of function that requires two different versions. And so this is conserved across over 300, 400 million years. And so there's something new that's come about that is not present in these other mall organisms. So then if you look, there's a paper from Mark Kay's lab that came out in 2017 and showed that a fragment of Lowe CAG1, but not a fragment from Lowe CAG, is actually required to enhance translation of the RPS28 ribosomal mRNA subunit. And so this is a hint of sort of some of the specialized function that one of those is helping control ribosome biogenesis and the other is not. Here's one more last example that I'll go over about I have so many examples of these, but here's one that's I think really important, the GlyGCC tRNAs. If you look at the human and mouse set, there are quite a few copies of these, but they're basically broken up into two major groups, the GlyGCC2s and the GlyGCC1s. And again, this is a base pair in the acceptor stem. And the only difference between these is for the GlyGCC2s, there's a UA at that 568 position. And in all the others, it's a GC base pair. Both of these should work fine in translation. We see plenty of other tRNAs with these kinds of changes, and so it's not obvious why we have that difference. However, again, if you look across not just human and mouse, but across primates, and again, this is a stand-in also for all mammals, insects and nematodes actually all have a CG at that position, and primates and mammals have a GC and UA at that position. Again, some kind of diversification that's been fixed and is continued for hundreds of millions of years. We do not know what the functional implication is. All we can see is that evolution is telling us now we have two versions of this tRNA that we maintain. So is there an expression difference between the two? Well, yeah, I probably wouldn't be bringing up if there wasn't. So for the GlyGCC2, that's kind of a housekeeping version of it, but GlyGCC1 is highly specialized and is very high in heart and skeletal muscle. There are other tRNAs that are specialized, but in this particular case, this one specialized version that is more recently evolved has some kind of specialized function in these tissues that we do not know the basis for these. All we can see is there's clearly strong evolutionary evidence and transcriptional evidence that these function and are regulated differently. All right, now switching to, how are we doing on time? Okay. Switching to human variation. We've accumulated a lot of information about variants in human genomes. And right now, if there are variants in human mRNAs and protein coding genes, there are predictors for what the impact is, whether that causes an early stop in the protein or a mistranslation or something like that. And you can actually get a readout from these different programs that are trained on thousands of genes and the sort of the functional implications of those. For tRNAs, there is no such guide. There's no indication or systematic way of judging if you have a particular variant in a particular tRNA, what the effect is. And so because of that, geneticists don't know what to do with those variants, so they have ignored them. It's frankly amazing that we have a gene family that is the oldest RNA gene family, probably. There are over 400 of them. They're highly conserved. We know that they're critical for translation of every single protein in our bodies, and yet clinical geneticists have completely overlooked them, again, because they probably didn't know what to do with them. So that's one of our missions of our lab, is to understand which variants are important and are tolerated. And you can look by seeing which variants appear in these genes and then which are absent. And so you can look at there are two main resources that connect medical records to full genomes. That's the All of Us database in the US and then the UK Biobank. The All of Us database, we've started looking at that and we have a lot of initial results. So the data we were able to pull over 30,000 single nucleotide variants across 531 tRNA genes, which included some pseudogenes as sort of as controls. And this is across over 414 genomes, 1,000 genomes. And we compared this and for verification with dbSNP, and we started with Nomad, which at the same tRNAs has 8,000 variants. So we can see more variants at more positions from this really rich set. So the goal is then to look for functional connection between gene variant frequency and some kind of biological importance. Now, in trying to figure out what the importance of any particular variant and change in the tRNA, there are lots of different functional areas of the tRNA that I've talked about briefly. The internal A and B boxes, these are in the D stem, which is on the left-hand side, and the T size C stem, if it's on the right-hand side. And so those are what are required for recruiting Pol3. So if there are mutations in these, that might impair transcription of that. Then, of course, you've got the anticodon, which is in the anticodon loop, and this is critical for proper recognition of codons and translation. Then you have identity elements that are required for proper aminoacylation by tRNA synthetases. And then there are other structural elements that help keep it folded properly and keep it stable and keep it from being degraded. So tRNAs are very complex in the different elements, and the number of these overlap each other. So we have in our program, we have something called tRNA Scan SE that we use to detect these, and we use them to evaluate them. So let me keep moving. So here on the bottom here is just an example of what the data look like for the All of Us database, the ARCH TCT4, which I've talked about. And we basically can annotate where we see variants and where we don't. So wherever you see it, sort of one of these white boxes, there's 0 variants in the population, which tells you that those are probably strongly selected against. And so, so this, in essence, is one of the most selected against genes that's selected against for variants in the population, even as heterozygous state. And so this is sort of is consistent with what we expected for this really important gene. Now, if you look across the different epi groups, and I'll try to wrap this up quickly, the constitutive ones have higher conservation, and I don't have time to go into some of these other measures, but the common versus rare ratio, if you look within a tRNA, you can look to see where the variants that basically are not highly selected for, because though they can change in tRNA without a lot of effect, versus ones that are highly selected against when you have variants like in the anticodon. And so the smaller this number basically means sort of the larger the contrast and this is an indication of the selective pressure. So as we kind of expected, the epi group A guys are the most selected against random variants across the tRNA, and then it goes up for these different groups. And I don't have time to talk about this Nochi score, but it's really fascinating how tRNAs haven't been able to be studied by these other methods that look for conservation or suppression of variants. Different parts of the tRNA are more highly conserved than the others. We can key on specific positions that are either have modifications that are essential for folding or for their function. And then, for example, the T loop has 2 selective pressures, both the B box as well as these other elements. And so I'll keep on moving, but basically every position has sort of a different importance, and they are slightly different between the different groups of tRNAs. And so we can tease these apart to look to see what the relative importance of any given nucleotide is and where they can vary and where they don't in populations. So one of the surprises here is that roughly half of these silent tRNAs are under strong purifying selection, half of them, almost 50 of them. And we still haven't found where they're used, but we know that they're important in human populations because variation at them is suppressed. And for the epi group C guys, a lot of these have purifying selection at the A and B boxes. They're internal promoters versus weaker selection against variants in the anticodon translation. This suggests that some of these are important actually as insulators, as chromatin elements, because recruitment of the Pol3 machinery to the A and B allows them to be insulator elements. All right, so I've sort of run out of time. So just in summary, tRNAs in mammals are extremely diverse. They've got all kinds of regulatory programs. The diversity of regulation that we see shows that if you knock out or have a dysfunction of one particular tRNA, others can't necessarily take up its place, even if they have the same anticodon. Specialized isodecoder features and functions are barely explored, and geneticists need to start looking at these variants. And finally, we have an ultimate guide to human tRNAs, which integrates basically all of this functional genomics data, the evolutionary data that people will be able to look up and actually start to get some leads as to the importance of each individual human tRNA, their role, and then which parts of the tRNA seem like they are most important. So with that, I'll acknowledge lots of people in the lab that have contributed to lots of different aspects of the tools that we use and the evolutionary analysis. And I'll leave you with this, the flyer for the RNA Futures meeting that we're all very excited for people to come to. Here's a QR link, and I will leave it at that. Thanks.
Great.
Any questions?
Thank you, Todd.
Sure.
There are a few questions in the Q&A. I'm not sure if we have time to go through all of them, but I know we can continue answering them, typing answers. I'll start with, is it known if differential expression of isodecoders across tissue contexts ever affects which synonymous codons in a given amino acid family are most optimal in a given context? Um,
Yeah, so the balance in every tissue is a little bit different between the different isoacceptor abundances. So, and those are often probably evolved and shaped towards the proteins that have to be sort of, are most important or specialized for those tissues. So, absolutely, for the tRNA abundances have a huge effect and are felt by and this is why we have differential codon usage, synonymous codons, I think.
Great. And we'll take one more. Have you looked at tRNA fragment expression in those different samples?
Yes, we have fragment expression from didn't have time to talk about, but we use OtterSeq. OtterSeq gives us both the full-length tRNAs as well as the fragments, as well as information on a large subset of the modifications in those. And so we are working through that very complex data, but when paired with all these other kinds of data, they're giving us a lot more clues than previously, most people were just scratching their heads as to why certain fragments are more abundant in some places than others. Okay.
Great. Okay.
All
There's a
right.
few more in the Q&A that I'll let you type answers to. And with that, I'll hand it over to our next moderator.
Okay. Thank you.
Hello, everyone. My name is José Casas. I'm a postdoctoral researcher here in University of Galway in Ireland. And it is my great pleasure to introduce our next speaker for today's seminar, Dr. Casas-Whysall. She's a senior lecturer in University of Galway and University of Liverpool, and she is the head of the Physiology department in the University of Galway. She earned her Ph.D. from the University of East Anglia in the UK, where her work focused on the role of microRNAs in skeletal muscle development. This research led to the significant discovery of several microRNAs, mechanisms that control PAX3 expression during embryonic development, which is a fundamental step in the understanding of how microRNAs regulate muscle mass and function. Currently, her research team is studying the therapeutic potential of both standard and oxidized microRNAs to combat muscle loss during aging and diseases such as cachexia and ALS. Furthermore, she is leading efforts to study BATPA's related conditions with the ultimate goal of developing innovative intervention and target RNA therapies. Please join me in giving a warm welcome to Dr. Whysall as she shares her insights into the oxidation of microRNAs in aging and disease. And please just remember to type all the Q&A and all the questions in the Q&A box instead of the normal chat.
Okay, thanks very much, Jose. Sure. Okay, so hopefully everyone can see my slides and hopefully you can all hear me. Okay, so welcome from Galway in Ireland. We are west of Ireland, right by the Atlantic Ocean, and we are the closest point to the US in Europe, apparently. So you can nearly see us if you look through the window if you're on the East Coast. And we are part of a small cluster here in Galway RNA cluster. You can see our activities on the website. So today I'd like to focus a little bit about microRNAs and how they get oxidized during aging and disease. We are a lab who's disease focused rather than molecule focused, and particularly we are looking at muscle and neuromuscular degeneration during aging, as well as different conditions. This is important because as we age, we are all going to have at some point sarcopenia, which is loss of muscle mass and function, and that can affect quality of life. But muscle wasting is also present in other conditions such as ALS, sepsis, critical illness, or cachexia. And there's more and more data to show that general outcomes of the treatments for some of these conditions actually are associated quite a lot with muscle health. And currently, there isn't really a treatment for lost muscle. Nutrition and exercise remain the best approaches. And this is probably associated with the complexity of muscle wasting. A lot of like with other conditions, what happens is that the expression of multiple genes is disrupted. And we think that microRNAs come into play here because they can regulate multiple genes at the same time. And that through this, they can regulate specific processes. And in a maybe not as chaotic way as the butterfly effect, if you change the levels of one microRNA, perhaps you can regulate the state of a disease. I probably don't need to remind this audience about the Nobel Prize in Medicine in 2024. And just a brief reminder, we focus on the canonical function of the microRNAs. So this is where they recognize their target sites within the mRNAs. And that leads to mRNA destabilization, degradation, or translation inhibition. And we and others have shown through different approaches that if you knock out dicer, for example, which is responsible for processing of these, you either get delayed myogenesis, premature muscle wasting, or defective muscle regeneration. So I've been studying microRNAs for quite a while now. And when I started, we used to look through papers or microarrays, try to select microRNA candidates that are basically changing, their levels are changing in specific conditions. We then moved to slightly more sophisticated computational modeling. And now in the lab, we were lucky to work with a data scientist, Karen, who was able to use machine learning and take all different RNA-seq data sets, build a model of muscle aging, and layer onto it some microRNAs. So in a kind of divine way, we actually found some of the microRNAs that we have been studying in the lab, but also some new candidates. So one of these is microRNA 181, which we have shown that regulates muscle mass and strength during aging through regulation of mitochondria. And you can see here, if you upregulate this microRNA, or actually what we do is deliver synthetic microRNA into the mice through intravenous injection, their muscle function, which is force, you can see here in old mice particularly, they lose muscle mass and function. It's restored. And this happens through kind of kickstarting mitophagy and regulating mitochondria. And you see mitochondria as something that comes across a lot when we talk about muscle. Maria, who's present on this call during her PhD, characterized the role of microRNA 379 in cancer cachexia in Lewis-Lung carcinoma model, where she found that this microRNA, through regulation of, again, mitochondria and inflammation, can preserve muscle size in cachectic mice. And we've also shown before that another microRNA 143, again, regulates senescence of satellite cells, and that's through regulating of peroxyredoxin 6, antioxidant signaling, and again, mitochondria. So we started looking a little bit at mitochondria and oxidative stress, and we worked very closely with Dr. Brian McDonough here in Galway and before in Liverpool. And oxidative stress is something that happens during aging and disease a lot. Muscle also produces ROS during exercise, but that's regulated in adult and healthy organism. And what has been shown by loads and loads of different papers is that these reactive oxygen species can actually modify different molecules within the cells. So DNA damage is very well known, very well associated with oxidative damage, and it's known to cause mutations as well as some diseases such as cancer. Lipid peroxidation can lead to cell membrane damage, but also cell death through ferroptosis. Protein oxidation, again, can lead to loss of function, misfolding, and so on. So RNA actually has been shown to be oxidized as well. It has been shown in different types of disorders. This is mainly mRNA, rRNA, and tRNA. Particularly here, you will see some of the neuromuscular disorders, but also in muscle. So the work from Esther Dupont-Verstegen in Kentucky has shown that in muscle of people, these are rats, of all rats, you will see more oxidized RNA, particularly after inactivity, than oxidized DNA. So it seems that RNA may actually be a better marker of kind of ROS damage within the cells. But is it more than a marker? So here is the structure of the RNA if it gets oxidized at G. I'm not a biochemist. I'm kind of a cellular biologist. So this is not my model, but I trust G is the most oxidized nucleotide, and that has been shown by different people within the RNA. And what it means is that it may pair with C still, but actually it can also pair with A, which for microRNAs could be very important. So this simplified schematic kind of shows some of the possibilities if Gs get oxidized within microRNAs, particularly within the seed sequence, which is really important for targeting of mRNAs. Maybe the microRNA can no longer bind to its target, and therefore is no longer to regulate the expression of these genes, or maybe microRNA with oxidized G binds to alternative mRNA targets, and there's a few publications to show this, and targets different genes. Or maybe it's a mix of both, or maybe it's neither. So this is what we have been trying to figure out. First, we wanted to see if we can actually detect oxidized microRNAs in muscle during different conditions. So these are some of the data sets we collected throughout the time. And we have taken two approaches. This is a few years back, so there's alternative approaches available nowadays as well. But when we started, we isolated a small proportion of RNAs and either sequenced those or did IP using antibody to OXOG, and then sequence the proportion, this proportion as well. And what we started to look at is changes in the levels of microRNAs in the two fractions enrichment. So how much of the microRNA that was pulled out with the IP, what proportion of it forms of the native microRNA. And we also started looking at what actually changes within the sequence we can see. So can we actually see this G2G signal in the RNA sequencing? And again, because we had a few data sets, we thought, okay, is there anything common in the microRNAs that we consistently see as enriched in the oxidized fraction? So a lot of them show a signal in this G to T transversion in the seed sequence, and these are some of the positions. I have to say, though, we don't normally see just one G being oxidized, so I think this is important. And it seems that there is a mix of different Gs being oxidized for some of the microRNAs as well. So Karen was able to use machine learning on these data sets to find different features of the microRNAs that are consistently oxidized. And I think not surprisingly, the most common element between them was that they are enriched in G bases. And actually, they have a lot of Gs within the seed sequence. But there is no link with their abundance or the expression levels, and also not their turnover. So then we ask if there are any functional consequences of microRNA oxidation within the cells. And in our lab, we use in vitro model cells as well as in vivo models, mice. So here, what we did is we identified the microRNA 378, which is downregulated in muscle of mice and humans during aging. And there are some publications to show its links with muscle mass and strength, including in humans and also exercise. So this microRNA is downregulated in aging. And what we did is we delivered synthetic microRNA or antagomere into adult or old mice. And we saw that with microRNA treatment, we were able to restore a little bit muscle mass in the old mice. And that was also reflected by the size of the myofibers. And we also saw changes in muscle force. This microRNA was also one of the detected as oxidized in different conditions, including human aging. And here you will see also microRNA 184, which has previously been shown to be oxidized in the heart. So it was kind of like a positive control for us to see it in our data as well. So instead of delivering oxidized microRNA to the mice for different reasons, we decided to try and based on the position of the oxidized Gs within the microRNA, design a molecule that will bind more preferentially to the microRNA that is oxidized than the one that's not, and deliver that into the mice. So in adult mice, this antagomere to oxidized 378, there's different caveats associated with this, didn't really show any effects, as you'll see here. This is a section of a muscle stained with WGA. There's no change in fiber size or muscle mass in these mice. But in old mice, we saw that there was a slight shift towards higher fiber size, bigger fiber size, but also the mice maintained their force during old age. Not to the same level as microRNA 378. The change was a bit smaller, but there was improvement. So what we did next also is not shown here. We looked at different targets of this microRNA 379 or changes in gene and protein expression in mice treated with antagomere oxy 378. And again, mitochondria-related pathways came up, and we saw quite a nice distinction between the targets of microRNA and the oxidized microRNA. And I'm happy to talk about this more during questions. So we also tested another microRNA, so oxidized microRNA 133. So here it didn't actually come as one of the most enriched microRNAs. This microRNA is really highly expressed in the muscle, but consistently it was detected to be oxidized in different conditions, including aging and cachexia. And so Maria, who's on the call, did two sets of experiments. One where she took adult and old mice, similarly with microRNA 378, and treated the mice either with microRNA 133, particularly the old ones, to restore the levels of microRNA in aging, or antagomere to oxidized 133, again based on where those oxidized Gs are within the sequence, or combination of both. And we added this time in the adult mice a group with oxidized scrambled. And so scrambled is just a microRNA that is it doesn't exist in the mouse genome, hasn't been detected. It's a scrambled sequence of the microRNA 133, so shouldn't in theory have any effects, and then oxidized microRNA 133. And what we found is that the oxidized scramble, for some reason, had more negative effects than microRNA 133 in old mice when we looked at grip strength or fiber size. And in old mice, on the other hand, we were not able to see restoration of either grip strength, which is a proxy for muscle strength, or fiber size when we overexpressed microRNA 133, but we saw some effects of the antagomere oxy 133 or the combination of both. And similarly, in cancer cachexia, in a male or male or female mice, we saw some changes again. So here the mice were either control cachectic or cachectic with antagomere to oxidized microRNA 133. And so this is the yellow one. You see the cachectic mice get weaker, and this was slowed down to some degree by the inhibition of the oxidized microRNA 133, particularly in male mice. There were very limited changes in fiber size, and when we again analyzed genes that could be regulated by either microRNA or the oxidized microRNA, we found that microRNA 133 had quite a specific signature, an enrichment of genes, which is consistent with what has been shown in the literature before. The oxidized microRNA 133 had really big overlap with genes regulated by microRNA 133. So there was definitely not the same specificity as we've seen for 378 or in the published data. And there's quite a lot of organelle-enriched terms. And we've looked at other microRNAs as well. So in total, we looked at 6 different microRNAs and their oxidized counterparts, as well as their scrambled controls and oxidized scrambled controls. And we found that among these, there seems to be emerging a pattern where some signaling pathways within the cells seem to be dysregulated upon interaction of these oxidized microRNAs or their scrambled controls. So we are now wondering whether just simple change in gene regulation by the microRNAs is actually the only mechanism. In fact, it's not always very obvious. We found microRNAs which, when you oxidized them, target different set of, or largely different set of genes than the microRNAs. The counterparts non-oxidized microRNAs, but also some microRNAs that actually the overlap between the genes regulated in the cells as determined by RNA-seq is very quite big between the microRNA and its oxidized form. So there are a few papers now to show that, but this is particularly important one, which shows that microRNA 1, another microRNA that's expressed really highly in the muscle and the heart, it's oxidized specifically in position 7, and that changes quite neatly the set of genes it's targeting. On the other hand, we have this literature showing that actually, if you have this now is not microRNAs, this is mRNAs, but what they have shown is that if you have an oxidized RNA, actually what happens is the damage kind of continues and you start to detect A-basic sites. And perhaps this is something we're missing here in our data, so we're going to look at that a little bit closer. And maybe, and this seems to be what our data is pointing us to, is that the story is a little bit more complex than very neat position-specific oxidation and very neat mistargeting of genes. Maybe it's a combination of dysregulation of targeting, but also activation of some different pathways that may be a result of production of these A-basic sites. There are oxidized RNA in general, oxidized RNA binding proteins within the cells. There are different cold shock proteins within the cells that could be some candidates to explore. So I will leave this as an open question and happy to take more questions, but thank all the lab for their hard work on this project that has had a lot of obstacles. So thanks very much, and thanks for your attention.
Thank you, Kasia. So I'm gonna start reading the questions in the Q&A. So we have a question, and is there a sex-specific effect of microRNA 378? And is there a difference between mated and non-mated females?
Yeah, so for 378, this is quite an older study, so we only used ever males. But for every other microRNAs that we have studied, there's a big effect of, there are big sex differences. We only study non-mated females, so we never actually looked in mated females. But undoubtedly, in the muscle tissue, there are big sex differences.
Okay, so then the next one is, does microRNA oxidation happen more frequently in cancer as well?
Yeah, so we found that if we look at aging, which is kind of chronic low-level inflammation and oxidative stress, compared to a scenario like ICU or cachexia, there's a big difference. We see more microRNAs with higher enrichments in those conditions like cachexia or ICU scenarios.
Okay, and I think we have more time, so I'll ask, do you see switching in targets between normal and oxidized microRNA 133? Especially, are there any new targets in the oxidized one?
Yeah, so we see different genes regulated, but we haven't been really able to match the sequences of those regulated genes to the microRNA. So I'm not really sure that they are targets or if it's something else that's happening within the cell. And microRNA 133 is the one when we started thinking about maybe it's not just target dysregulation. So I am not sure there are very clear new targets for this microRNA.
Perfect. So that's all of the questions.
Perfect. Thanks very much.
Yeah, so with that, we will close the seminar. The next RNAi collaborative seminar is in June 10th at 4 p.m. and will be hosted by Yale Center of RNA Science and Medicine. Thank you very much, everyone.